Hi everyone, this is going to be the fourth video on the topic of biochemistry and molecular biology. In this video we're going to be looking at perhaps one of the most important classes of biological macromolecules, the proteins. So let's start by looking at the significance of proteins. Now, the significance of proteins in biological systems really can't be overestimated. They're connected with all phases of the chemical and physical activities of cells, tissues, organs and organ systems. Now, the function of proteins are related to their specific shapes, and so some of them will act as enzymes, others might be hormones, antibodies, they can be transporters for oxygen and electrons, they can also form structural elements like skin, hair, feathers, nail and cartilage. And so proteins are incredibly important, and they tie in with what's called the central dogma of biology, which is something we're going to explore in later lessons. So, the building blocks of proteins, as we mentioned in the first lesson in this topic, are the amino acids. Amino acids are the monomers of protein polymers. And so, proteins are large molecules. They're very, very large as molecules go, and they have these very complex three-dimensional shapes. So, each amino acid, as we said before, has a basic structure, and the key elements of that structure are shown as follows. So, we have what's called the alpha or central carbon, surrounded by four individual single covalent bonds. One of them is to an amine group, or an amino group as it's sometimes called, and the other one is to a carboxylic acid or carboxyl group on the other side. Now, the remaining two bonds on the alpha carbon, one of them is to a hydrogen atom, and there's also a bond to a variable group. Now, all amino acids have this general structure that you're seeing here, but it's the variable group that causes them to differ from one another. And so all amino acids have this basic structure, but it's the variable group which is different from one amino acid to the next. And there are about 20 naturally occurring amino acids, all of which have this general structure, but only vary between each other in terms of the variable group, or the R. Now, in a previous lesson, we looked at some of the examples of these in terms of three-dimensional models of a couple of different examples of amino acids. So just to remind you, even though this structure looks relatively straightforward, the way that these bonds are displayed in the structural formulae are not necessarily the way that the bonds are arranged in 3 dimensions. So let's have a quick look at some amino acids here. Now, this is glycine. Glycine is the simplest amino acid. The R group for glycine, the variable group for glycine, is simply one hydrogen. So we can see here the skeletal formula for glycine, where we have the amino group here, we have the carboxyl group here, we have the alpha carbon here with one hydrogen, and the other bond is to the variable group, which in this case is just simply a hydrogen atom. So this is glycine here. Now, in contrast, some of the other amino acids can have much larger, much more complex R groups. So if we take a look at, for example, arginine that you're seeing here, so this is the skeletal formula here. We can see that we have here the carboxylic acid functional group here. We have the amino group here. This is the alpha carbon, and we have one bond to a hydrogen, and the remaining group here, this very large linear looking group over here, this is the variable group for arginine. So you can see that the variable group for arginine is much larger and much more complex than for something like glycine. Now, again, in contrast, some of the variable groups are both large and cyclic. So tryptophan, which you're seeing here, has a heterocyclic variable group. So again, what you're seeing here is the skeletal formula. Here we can see the amino group, which is represented here, and over here we can see the carboxylic acid functional group. Both of those are attached to the alpha carbon that you're seeing here. The alpha carbon has a single bond to a hydrogen, which of course, as we said before, is not represented in a skeletal formula, because skeletal formulas are essentially a shorthand way of showing organic structures, and so not all of the bonds to the hydrogen atoms are shown here. So this is the alpha carbon, and this group here, including this carbon and this heterocyclic structure containing a 5-membered ring and a 6-membered ring linked to each other with delocalized electrons in those rings, this is the variable group for tryptophan. And so these are the variable groups that you can see, which vary quite differently from one amino acid to the next, going from the simplest glycine to the more complex. Okay, so the variable group is what differs from one amino acid to the next. And so we have a large variety of amino acids, and there are about 20 different naturally occurring amino acids. Now, individual amino acids display a tetrahedral organization of the groups around the central or alpha carbon. Now, the reason for this is that carbon is an atom which forms single covalent bonds around its atoms in a tetrahedral orientation because there are 4 single covalent bonds and no lone or non-bonding pairs in the outer shell of the carbon atom. And so all of these individual covalent bonds, because they are regions of negative charge density, essentially a covalent bond is a pair of shared electrons which causes an electrostatic force of attraction between the 2 nuclei. Well, what you have here between these 2 groups or these 2 atoms is a region of negative charge density, and the same thing for all of these 4 other bonds. And so the 4 single covalent bonds around this central alpha carbon in this amino acid molecule will repel each other, and they do so equally. And so essentially they will organize themselves to maximize the bond angle between each other, and this results in this tetrahedral orientation. And it's called tetrahedral because the 4 groups around the central carbon atom are placed in a position where you would find the vertices, the corners of a 4-sided 3-dimensional shape, a tetrahedron. So individual amino acids will display a tetrahedral shape due to the angles of the bonds between the atoms connected to the central carbon atom. Now, as I said before, there are 20 different naturally occurring amino acids, and they differ from each other by virtue of the R group. So the simplest of those is glycine that we saw before. Again, glycine is this one that we're looking at here. So again, we can see here we have a central carbon, and as you can see, the 4 bonds surrounding that central carbon atom are arranged in a tetrahedral orientation relative to each other. So this is the simplest of those 4 of those amino acids, those naturally occurring amino acids, where the R group is simply a hydrogen atom. So if we take, for example, alanine, alanine is an amino acid where the R group is simply a methyl group. So we have the amine. Now, amine groups are basic in the sense that they will accept or bond with protons, whereas carboxylic acid functional groups are acidic because they can release protons or hydrogen ions into aqueous solution. And since amino acids possess both an amine basic group and a carboxylic acid acidic group, it means that overall the amino acid is a neutral molecule. So here's another example of an amino acid. Here we can see that in this case the R group is a CH2 group connected to a carboxylic acid functional group. And so in this case, this amino acid has 2 carboxylic acid functional groups, and this makes it an acidic molecule because you only have 1 basic group and 2 acidic groups. In contrast, this is the R group that we saw. This is the R group for a different amino acid that we saw before, and this is a basic molecule because you have 2 basic amine groups and 1 carboxylic acid functional group. So this goes back to the definition of acids and bases as being proton donors and proton acceptors respectively. So acids are defined as proton donors, or substances composed of molecules that dissociate and release hydrogen ions or protons into aqueous solution. And the hydrogen attached to this carboxylic acid functional group can detach itself in aqueous solution. Carboxylic acids are generally weak acids. They dissociate weakly in aqueous solution. But if you have 2 of those versus 1 basic amino group, then overall the molecule will be acidic. Now, bases are defined as proton acceptors. And the reason that an amine group is has a basic nature is because there is a lone or non-bonding pair of electrons on the nitrogen in these amine groups. And that lone or non-bonding pair of nitrogens can form a dative covalent bond with a hydrogen ion when that hydrogen ion approaches closely enough to this group. And so if you have 2 such groups versus 1 carboxylic acid functional group in an amino acid molecule, then that molecule will be basic. So another important type of variable group that you need to be aware of, and we'll be discussing later, is the variable group that you're seeing here, and this is in the amino acid cysteine. Now, this variable group contains a sulfur atom, and this is going to be rather important because there are only two sulfur-containing amino acids. These are cysteine that you're seeing here, and another amino acid, methionine. And the reason that they're going to become important a bit later on is we're going to be talking about the ways in which the amino acid R groups, when the amino acids are bonded together in a polypeptide, they can actually form bonds with each other. And one of the more important, stronger bonds that can be formed from adjacent amino acid residues, the R groups, is the disulfide linkage. So we're going to be talking not just about the individual amino acids, but the ways in which they connect with each other chemically to form polypeptides, and then the way those polypeptides eventually form functional proteins. So we need to be aware of the levels of protein structure. So protein molecules are made up of many different amino acid monomers linked together, and amino acids are joined together by condensation reactions which involve the removal of a water molecule. And when you join two such amino acid molecules together by a condensation reaction, you form what's called a dipeptide, and then by successive condensation reactions you can build up a chain of amino acids from tripeptides to polypeptides, and ultimately the polypeptide will form a protein. Now, we covered this in a previous lesson, but it bears repeating here. So two amino acids can join together by a condensation reaction. This always occurs by the removal of a hydrogen atom from the amino group of one amino acid and the oxygen and hydrogen from the carboxyl group of an adjacent amino acid. And so this would involve the removal of a water molecule, and this then forms an amide or peptide bond. Now, in biology we call this a peptide bond because it is found in polypeptides, but in a purely organic chemistry context you would refer to this as an amide bond or an amide linkage. So the amino acid sequence, the specific sequence of amino acids in a protein, in other words the linear sequence and the identity of those amino acids in that linear sequence, constitutes what's called the primary structure of a protein. So this is formed by repeated condensation reactions between many amino acid molecules, and this is all catalyzed by a series of enzymes. Now we're going to be looking at the formation of polypeptides and the mechanism by which the information in the base pair sequence of DNA codes for this through transcription and translation in a later topic. But for the moment, it's enough to understand that the primary structure of the protein refers to the number, the type, and the sequence of amino acids that makes up this linear chain, together with the peptide bonds that hold them together. Now, different proteins have different primary structures, and so different proteins can be said to be made up of different types, numbers, and sequences of amino acids, making up the primary polypeptide chain. So the specific protein which is made and its function will depend upon a number of factors: the types of amino acids it contains, the sequence in which those amino acids occur along the molecule. And so here we can see two different polypeptides. They have the same amino acids in the chain, but those amino acids are present in a different order. And this will result ultimately in those two proteins having very, very different three-dimensional shapes and therefore different functions. Now, this seemingly small difference could actually be cause a very large difference in their overall structures and in their functions. And so, for example, this small difference could be all that's different between pig and human insulin. So ultimately, the sequence of amino acids in a specific protein is coded for by the sequence of bases referred to as the triplet code in the DNA, which makes up the gene. So again, we're going to be looking at molecular genetics and the way that information is encoded in DNA, which then leads to the expression of that information as protein structures in a later topic. So the primary chain of amino acids in a protein, ultimately, because you are chaining together those amino acids by taking the amino end and the carboxyl ends of two adjacent amino acids and forming a peptide bond between them, it means that no matter how many amino acids you link together with peptide bonds, you are always going to have an an amine end of that polypeptide chain with a free amine group at one end, and you're always going to have a free carboxyl or carboxylic acid functional group at the other end of that polypeptide. And so we refer to these as the N-terminus and the C-terminus. Now, the secondary structure is determined by the spatial arrangements of the atoms that form the backbone or the linear chain of the protein. And so, in a sense, it's the specific sequence of amino acids in the primary structure which goes on to cause the secondary structure of that protein. And so the amino acid chain, the polypeptide chain, the primary structure, ultimately can coil and also fold into secondary structural motifs, things like helices and sheets. And so these can be referred to as alpha helices, or other shapes like beta pleated sheets or beta barrels. These are referred to as the secondary structural motifs, or the secondary structure of that protein. Now, the helix and the beta pleated sheet shapes are secondary structures, and these diagrams give you a sense of what those look like. And so what we're seeing here, the blue line being shown here, the kind of ribbon-like line, represents the linear sequence of the polypeptide, but that linear sequence is now folding into three-dimensional shapes. In this case, an alpha helix, in this case, what's called a beta pleated sheet. And these beta pleated sheets can sometimes curl around on themselves to form what's referred to as beta barrels. So the amino acid chain will coil into right-handed helices, and hydrogen bonds will form between the oxygen and the hydrogen atoms that have been brought into close proximity by that coiling. And so this is called an alpha helix. So what you're seeing here, these red lines that you're seeing here, are the hydrogen bonds. And so the hydrogen bonds help to stabilize the secondary structure. Now, hydrogen bonds are not the same as the covalent chemical bonds between the atoms in this polypeptide chain. Those are much stronger bonds. Hydrogen bonds are examples of what's referred to as physical bonds or intermolecular forces. Now, in general, intermolecular forces or physical bonds are much weaker forces of attraction than covalent chemical bonds. They require much less energy to overcome or break. But of all of the types of intermolecular forces, hydrogen bonds are in fact the strongest type of intermolecular force. And so it's the formation of hydrogen bonds between the amino acid residues in this polypeptide chain which causes this alpha helix, this right-handed helix, to be stabilized. So another example of a secondary structural motif that we saw is the amino acid chain folding back on itself to form parallel chains, and this is how a beta pleated sheet can be formed. Now again, it's the oxygen and hydrogen atoms that have been brought into close proximity with each other, forming hydrogen bonds with each other. Now a hydrogen bond is essentially an electrostatic force of attraction between the lone pairs of electrons on oxygen atoms and the unshielded hydrogen atoms in other parts of a molecule or between two molecules. Now, again, this is probably more familiar to those of you who've studied A-level chemistry, but it is required knowledge for A-level biology as well. So a hydrogen bond is, in order to have a hydrogen bond, you need a couple of things to a couple of sets of conditions. So an example of a simple substance which has hydrogen bonding between its molecules is water. Now we're going to be discussing water in a later lesson of this same topic, but for now it's important to understand that hydrogen bonds are a relatively strong form of intermolecular forces which usually can form between highly electronegative atoms like oxygen, which have lone pairs on them, and hydrogen atoms in other parts of the molecule which are bonded to other atoms. And so what you have here is the oxygen atoms in the carboxylic acid functional groups bonding with the hydrogen atoms in the amino functional groups in very disparate parts of this this polypeptide chain. So for example, here we can see that the polypeptide chain has curled round on itself, and at this particular location, the amine functional group here on this amino acid residue has bonded to the carboxylic acid functional group on this amino acid residue, even though those two amino acid monomers are separated from each other by several intervening amino acid monomers here. So this is how a beta-pleated sheet forms. So both alpha helices and beta-pleated sheets are secondary structural motifs which are stabilized by hydrogen bonding between the atoms on one amino acid monomer and the atoms on another amino acid monomer. So these are all hydrogen bonds, and these help to stabilize this structure. So the tertiary structure is a higher level of organization again, and the tertiary structure is essentially the three-dimensional structure of the protein once all of those different regions of the protein containing alpha helices, beta-pleated sheets and other structural elements fold up into a three-dimensional what's called a globular structure. So all globular proteins display tertiary structure, and once the secondary structure is formed, the molecule then bends and folds further into a more complex three-dimensional shape containing all of those structural motifs, the alpha helices, the beta sheets and so on, all arranged in a particular manner. So myoglobin, this diagram represents a myoglobin molecule. So myoglobin is a globular protein which is found in muscle cells, and its structure actually resembles each of the four subunits found in a hemoglobin molecule, and it performs a similar function. It binds and unbinds oxygen in the muscle cells itself. So myoglobin is a globular protein, and this tertiary shape is the highest level of structure found in myoglobin. And again, within the tertiary structure, there are a variety of bonds, hydrogen bonds and also other types of bonds, which help to stabilize this very specific three-dimensional structure. Now, hemoglobin, in contrast to that, displays an even higher level of protein organization called quaternary structure. Now, quaternary structure is the level of protein structure where you have separate tertiary structures of separate polypeptide chains all coming together, forming a complex with an even more complex structure. And so there are actually 4 polypeptide chains, each of which has its own tertiary structure, which have all been combined together into one molecule of hemoglobin. So quaternary structure is a level of protein structure displayed by proteins that consist of more than one polypeptide chain. So hemoglobin is a protein which displays quaternary structure, and hemoglobin consists of 4 polypeptide chains, the 2 alpha chains and the 2 beta chains, and these are held together by relatively weak intermolecular forces referred to as van der Waals forces, or instantaneous dipole-induced dipole interactions. So each polypeptide chain contains an ion-containing heme group, and this ion-containing group is the part of the molecule which binds and unbinds oxygen. Now again, this molecule will come up again in our discussions in a later topic on transport, where we look at the way that the hemoglobin molecule actually works, but for now this is a good example of a quaternary structure, where you have separate tertiary structures, separate globular proteins all coming together and being bound together into one functional unit. So let's have a closer look at the kinds of bonds that can help stabilize both secondary and tertiary structure. So as the chains of the amino acids bend and fold to form secondary and tertiary structures, there's various types of atoms which are brought into close proximity. Now, when those atoms are brought into close proximity with each other, they can form a variety of bonds, as are being shown in this diagram. So let's have a look at some of these. So firstly, hydrogen and oxygen atoms, like these, can form what's called hydrogen bonds. Now, these are a type of intermolecular bonding. They are not covalent chemical bonds. What's happening here is one of the lone pairs on this oxygen atom is forming an electrostatic force of attraction with the proton, the hydrogen nucleus, sitting at the end of this covalent bond here. Now, again, hydrogen bonding can occur whenever you have a highly electronegative element such as oxygen or nitrogen or fluorine, for example, which has lone pairs on it, coming into close proximity with a bonded hydrogen nucleus. Now, the hydrogen nucleus sitting here donated its only electron into this covalent bond, sharing that electron with an electron from the nitrogen atom. And so the hydrogen nucleus doesn't have other shells of electrons orbiting it, and represents a very dense region of positive charge sitting at the end of this covalent bond. And so In contrast, this highly electronegative oxygen atom has a couple of lone pairs, non-bonding pairs of electrons sitting here in its outermost shell, and those non-bonding lone pairs represent a very dense region of negative charge. And so this very dense region of positive charge and this very dense region of negative charge will electrostatically attract each other if they're brought close enough together, and that electrostatic force of attraction is called a hydrogen bond. And so this is one of the most important types of bonding that can stabilize both tertiary and secondary structures. And so this is another example here of a hydrogen bond. Here we're seeing a different group of atoms forming a hydrogen bond. Here we see the OH group from either a variable group of an amino acid here, or let's say the carboxylic acid functional group of the terminus of a particular polypeptide chain. And this can form a hydrogen bond with, again, the carbonyl oxygen of some other amino acid residue somewhere else. So these are hydrogen bonds. Now, the R groups of two amino acids which contain sulfur atoms, like cysteine and methionine, can form a very strong covalent bond between those two sulfur atoms. Now, in contrast, this is a chemical bond. It's a bond between atoms involving a shared pair of electrons. And in contrast to a hydrogen bond, covalent chemical bonds like these are much, much stronger. They require a much higher level of energy to break. And so these are referred to as disulfide bridges or disulfide bonds. Another type of interaction is an ionic bond, and so many of the carboxyl and amine groups which are found in both the termini of amino acid or polypeptide chains, and also in some of the variable groups, when they are in aqueous solution, they will dissociate and ionize. And so a carboxylic acid functional group might lose its hydrogen because it's donated that hydrogen, being an acidic group. And in contrast, the amine functional group of another residue may have bonded to a proton and formed an NH3+ group. And so these are groups or parts of the molecule which have discrete full negative or positive charges, and if they're brought closely enough together, like any two oppositely charged ions, they can form a very strong ionic bond. And so ionic bonds, like covalent bonds, are strong chemical bonds. They are bonds essentially between atoms, in this case between ions, atoms that have lost or gained electrons. And so this type of bond is, compared to a hydrogen bond, very, very strong and very difficult to disrupt. It requires a large amount of energy to overcome. Now, many of the hydrophobic R groups, the hydrophobic variable groups, in other words, variable groups that contain arrangements of atoms which do not dissolve particularly well in water and are therefore hydrophobic, they can form types of intermolecular bonds which are called hydrophobic interactions. And so this is another type of electrostatic attractive force which can hold together disparate regions of that polypeptide structure, or even separate polypeptides. And so these are the kinds of interactions, the kinds of bonding that can stabilize and cause secondary and tertiary structures. So to summarize, the primary structure of a protein is simply the specific sequence of amino acids found in the polypeptide chain. This then folds into alpha helices and beta sheets, which are regular shapes, regular geometries found in substructures. This is the secondary structure. Those secondary structures will then further fold and arrange themselves into tertiary structures, forming what's called a globular or three-dimensional structure. And you can have multiple such tertiary structures bonded together occasionally to form quaternary structures. And again, an example of that would be hemoglobin. So you need to also be aware of how we test for the presence of proteins, and so this is done via a procedure called the Burette test. So to do this, a solution of the material which is to be tested is placed in a test tube. We add a little bit of sodium hydroxide to clear the solution, and a small amount of weak copper sulfate solution is then added down the side of the tube. And so if a protein is present, we get a purple-violet colour, and this indicates the presence of a protein. So this is the Burette test. Now, one of the most important functions of proteins is their role as enzymes, as biological catalysts. Now, there's going to be a separate lesson on the subject of enzymes later on in this topic, but for now it's enough to mention that enzymes are biological catalysts. They control various reactions in cellular metabolism and catalyze the hydrolysis of food materials during chemical digestion. So there's a couple of different representations of, for example, lysosome enzymes called lysozyme. So this would be one such representation where you are seeing the abbreviated names of the individual amino acids in the primary structure. Here would be a different kind of representation. This is a three-dimensional model, and here you can clearly see regions where the primary structure is folded into alpha helices, and also a region where you're seeing a beta sheet here. So there are also regulatory proteins. Now, hormones are involved in the regulation of cellular metabolism. So insulin and glucagon, for example, are both protein hormones that regulate blood glucose levels. And so another important function of proteins is as regulatory molecules like hormones. And so we're going to look at the specifics of blood sugar regulation in a later topic of this advanced level biology course on sensitivity, control and coordination. But again, it's important to know that proteins can act as regulatory, they do have a regulatory function, and this can be in the form of hormones. It can also be in the form of neurotransmitters. Oxygen transport, obviously, we've mentioned hemoglobins, and hemoglobin is incredibly important in mammals, for example, and also in other organisms. So hemoglobin in mammalian red blood cells transports oxygen to and from the tissues, and myoglobin is a similar molecule to one of the protein subunits found within hemoglobin, and this is found in muscle cells. Another important function of proteins, which again we will explore in the cells topic in more detail, is their function in membrane transport. So the membranes of cells and organelles will have protein transporters embedded within them, either extrinsic or intrinsic proteins, and many of these can function as channels, as carrier proteins, and as pumps, so-called protein pumps. And so all of these are important in simple diffusion, in facilitated diffusion, and also in active transport. Now, another function that proteins have, which is rather important, is the storage function. So ovalbumin is the type of protein found in eggs which stores the necessary proteins for the organism developing inside the egg to develop. Casein is a storage protein found in milk. And obviously the other function of proteins is their structural function. So collagen is the structural protein found in bones and connective tissue. So what you're seeing here is a micrograph of bone tissue. Here you can see an electron micrograph of individual collagen fibers, and these are made up of protein. Here we can see the horns of these two rhinos, which is made up of yet another protein called keratin. So keratin is a fibrous structural protein, and this protects epithelial cells from damage or stress. We also have keratin in our bodies, and rhino horns consist of pure keratin. So a human hair that you're seeing here in this scanning electron micrograph is made up of keratin, and so you find keratin in hair, nail, claws, hooves. It also forms a covering around horns. Now, horns like these, the central core of the horn is made up of live bone tissue, but the outer covering is made up of layers of keratin. Keratin can also form a different structure called B-keratin, which contains beta-pleated sheets. Now, this is the structural protein of reptilian scales, claws, shells, and also feathers, beaks, and claws of birds. So here we can see a micrograph, an electron micrograph of a feather, and here you can see the keratin and the way that it's arranged. Now, another function, structural function of proteins is their contractile and motile function. Now, You find contractile and motile proteins in muscles, for example. So in the topic on sensitivity, coordination and control systems, we're going to have a lesson specifically on the way in which muscular contraction is produced. And this is produced by protein fibers in your muscle cells of two different varieties, actin and myosin, and other proteins like tropomyosin. And they have this ratchet mechanism where the proteins literally ratchet against each other in order to contract the muscle. And so this is something that we'll explore in another lesson, but actin and myosin molecules are the contractile proteins of muscle tissue. And this is obviously a very important function in multicellular organisms like ourselves. You also have intracellular contractile proteins involved in, for example, the mitotic spindle during cell division. Again, we're going to look at cell division in a bit more detail in a couple of topics. In the cells topic, we're going to look at mitosis and the cell cycle. And in that topic, in that lesson, we're going to look at the role of the protein filaments which are formed by which form the spindle fibers, and these are important in moving the chromosomes around the cell in such a way that the chromosomes are distributed, the genetic material is distributed correctly to the two daughter cells. And the type of proteins involved here include tubulin. Now, tubulin is a protein which makes up these microtubules, and eukaryotic cilia as well as mitotic spindles are composed of these microtubules which are made up of this protein tubulin. Now, there's also the proteins which make up the cytoskeleton, which is the system of fibers and microtubules which runs all the way through the cytoplasm and is responsible for intracellular transport and movement of organelles and substances and vesicles, and also, in the case of organisms like amoeba, for example, responsible for changing the shape of the cell membrane during endo and exocytosis. Now, there's also the protective functions of proteins, and so fibrin is the protein which is formed during blood clotting, and thrombin and fibrin are both blood clotting proteins. Now, thrombin is actually an enzyme that catalyzes the conversion of the soluble blood protein fibrinogen into fibrin when the blood comes into contact with oxygen, when the surface of the skin is cut, for example. And the blood clotting cascade is actually a rather complicated sequence of biochemical reactions which ultimately result in the formation of these insoluble fibers made of fibrin, and this then causes the clot to form. So proteins can form a protective function. Now, another protective function of proteins is their role as antibodies in the immune response. Now, again, in the cells topic, we're going to be looking at immunology in a lot more detail, but for now, it's enough to say that antibodies, which are sometimes referred to as immunoglobulins, these are the proteins of the immune system. Now, antibodies are manufactured by the B lymphocytes, and they then interact with specific foreign antigens which have gained entry into the body, and either will cause those antigens to be, or rather the antigens on the surfaces of pathogens will cause those antigens and those pathogens to be agglutinated and to be found and destroyed more easily, or just inactivated by the presence of the antibody. So antibodies are also proteins and serve a function in the immune response. Now, there's a number of other specialized proteins which are only found in certain contexts and in certain organisms, but these are also important. So, for example, Antarctic fishes have evolved a unique type of antifreeze molecule, which are modified proteins. They are glycoproteins. They're proteins with sugar groups added to them. Now, these proteins basically have the same function as antifreeze. They prevent the fluids and the blood of these organisms from freezing solid in the sub-zero temperatures which you find in the Antarctic. There are also other specialized proteins. So, for example, these mussels secrete a specialized glue to attach themselves very, very securely to the rocks on the seashore. Okay, everyone, well, that's going to do it for today's lesson. I hope that was useful to you, and I'll see you in the next lesson, which will be about enzymes. See you then.